Kombucha is a drink obtained from sweetened tea fermented by the so-called tea mushroom,
You want to put it in the jar? What are you doing?
otherwise known as Japanese mushroom or SCOBY, symbiotic culture of bacteria and yeasts. The drink is slightly sweet, sour, refreshing and sparkling. It has a color from light gray to dark brown. Mushroom is a gelatinous structure of white or light orange color, made of cellulose together with microorganisms living there in symbiosis. The color of the drink depends on the type of tea used for fermentation. Black or green tea and sugar are best suited for the production of kombucha. As a result of fermentation, various products are produced, the most important of which are organic acids, ethanol and carbon dioxide. During the fermentation process, the yeast present in kombucha produces extracellular hydrolases that transform the sugar present in the tea into monosaccharides. In the case of sucrose, these are glucose and fructose, which are metabolized to carbon dioxide and alcohol up to 2% in the final product. Small amounts of lactic and acetic acid are also produced. Kombucha is made in many regions of the world, so there are many ways to prepare this drink. For 1 liter of drink, 100 grams of sugar and 5 grams of tea are used. After brewing and cooling the tea, the whole is poured into a previously sterilized glass or plastic container and inoculated by inserting a small piece of mushroom and pouring about 200 milliliters of the drink from the previous fermentation. The vessel is covered with a cloth that protects against air pollution, put aside in a shaded place and fermented at room temperature. The drink is ready after 3 to 5 days. Product must be kept clean in order to prevent the formation of mold. During production, metal devices should not be used to prevent partial dissolution of the metal due to acids in the drink. The tea mushroom is not a fungus, but is made up of many species of bacteria and yeast in a homogeneous culture. Symbiotic microorganisms occur in a variable quantitative and qualitative composition depending on its origin. The most common are bacteria of the species Acetobacter and Gluconobacter. The main strain present in kombucha is Gluconacetobacter xylinus, which synthesizes cellulose. The dominant strains of acetic acid bacteria are Acetobacter xyleum, Acetobacter pasteurianus, Acetobacter aceti and Gluconobacter oxidans. The most common yeast species are Saccharomyces, Saccharomycodes, Schizosaccharomyces, Zygosaccharomyces, Decara, Candida, Torulaspora, Colecora, Pichia, Mycotorula and Mycoderma. The drink contains various ingredients depending on the microorganisms present and the raw material from which the drink was prepared. Various organic acids are present, such as acetic, gluconic, citric, lactic, malic, tartaric, malonic, oxalic, succinic, pyruvic, usnic. From sugars, sucrose, glucose, fructose and vitamins, B1, B2, B6, B12, C. Apart from that, 14 amino acids, biogenic amines, purines, fats, proteins are present, a small amount of hydrolytic enzymes, ethanol, carbon dioxide, phenols, polyphenols, minerals, anions and lactone of glucaric acid and many other metabolites of bacteria and yeasts.
Was mit Mandeln, glaube ich.
The microorganisms in kombucha metabolize substrates in a variety of complementary ways. Yeast synthesizes invertase that breaks down sucrose into glucose and fructose. At the same time, they use fructose and produce ethanol in the process of glycolysis. Acetic acid bacteria use glucose to synthesize gluconic acid, and ethanol is metabolized into acetic acid. During fermentation, the pH of the drink drops due to the formation of acids. For the first 8 days, the obtained product has the characteristics of a refreshing drink, then it is transformed into food vinegar. Due to the presence of sugar, it is not recommended for people suffering from diabetes. Due to the alcohol content of up to 2%, it is not recommended for children and pregnant women. The drink has a positive effect on the intestinal flora.